Good afternoon. I'm Dr. Ronok Zahan, Associate Professor, Department of Pharmacy, University of Rajshahi, Bangladesh. So today's my presentation title is Mechanism of Action of Novel Nickel Complex in Simultaneous Reactivation of the Apoptotic Gene against Ehrlich Ascites Carcinoma cells. We know cancer is considered as the most devastating disease in the modern era, which is caused by the abnormal cell proliferation in the body. And it has 6 hallmarks. That is immortality, produce go signals, override stop signals, resistant to cell death, angiogenesis, and metastasis. And therapeutic potential of metal complexes in cancer therapy has attracted a lot of interest because metal exhibit unique characteristics: redox activity, variable coordination modes, reactivity towards the organic substrates. These properties become an attractive probe in the design of metal complex, which selectively bind to the biomolecular target with a resultant alteration in the cells and mechanism of proliferation. And the discovery of cisplatin was a defining moment which triggered the interest in metal complex as a new potent lead for cancer treatment. And in our study, we have used nickel as a metal for the preparation of metal complex. And why we used dithiocarbazate as a ligand? Because it has immense interest to researcher, because S-benzyl dithiocarbazate ship bases comprises one of the most important classes of mixed hard soft nitrogen sulfur donor ligands, having 4 potential donor atoms. And the interaction of these donors with metal ions give complexes with different geometric properties. Generally, the bioactivities of the ship bases and its complexes depends on delocalization of pi electrons, non-planarity in molecules, compound packings, and hydrogen bonding, thereby making variations in their bioactivity. And this piece of research work is an attempt to find out novel, simple, inexpensive, and safe chemical agents with a view to formulate new anti-cancer drug. And the total aim of study was to assess the biological activity of the metal complex by cell viability test by MTT colorimetric assay to check anti-cancer activities, that is % of cell growth inhibition, mean survival time, % increase in lifespan, morphological changes of cancer cell, expression of housekeeping and apoptosis regulatory genes, DNA fragmentation assay, and to evaluate other parameters like hematological test, liver function test, kidney function test, toxicity study on different organs like liver and kidney. And this is the total flow work. And first we prepare and purify the metal complex and characterize it by physical measurement, UV, IR, mass, and X-ray data. And second step, we have studied the biological assay of the metal complex by in vivo and in vitro. Here we have used different types of experiment. And this is the simple preparation procedure for metal complex. First, hydrogen hydrate and carbon disulfide get together and produce potassium dithiocarbazate. This potassium dithiocarbazate then in presence of benzoyl chloride produce S-benzyl dithiocarbazate. This is called proligand. This proligand in presence of 2,4-dihydroxybenzaldehyde produce ship base. This ship base then react with metal salt and produce metal complex. Then I will discuss the result section. This is the physical character of the complex, and our complex are brown in color. Its melting point was 190 to 196 degree centigrade temperature, yield 57%, and it was diamagnetic in character. And this is the overall IR data, UV data, and low resolution mass spectrum data. And this is the X-ray data for our nickel complex. And from the data, we have constructed the structure of the complex. And our complex was novel complex. So we have got a CCDC number. And that was 1842899. And We have studied molecular simulation and root mean square deviation and root mean square fluctuation by Discovery Studio with 5 apoptotic genes like bugs, BCL2, caspase 3, cytochrome C, and FAS, and we have got promising result in all. And we have also checked the antihemolytic activity, and our nickel complex have shown a very negligible percent antihemolytic action. It was observed that our nickel complex induced negligible percent hemolysis, that is 2.51 to 6.90, which implies that the component shows antihemolytic activity. And for the in vivo activity, to check anticancer activity in vivo model, we have used switch albino mouse, and our tumor model was Ehrlich Ascites Carcinoma cell. And treatment start on second day after 24 hours of inoculating cancer. Treatment dose was 0.3 milligram per kg body weight and 50 milligram per kg body weight. And this dose, 50 milligram per kg body weight, was selected after doing LD50 experiment. And the treatment time interval 24 hours. And our nickel complex have shown the percent of cell growth inhibition 51.81% when dose was 0.3 milligram per kg body weight. But we have got 75.75% inhibition when the dose was 50 milligram per kg body weight. And this is the another parameter for anti-cancer activity checking, that is mean survival time. And our nickel complex for 50 milligram per kg body weight, it have shown 28 days when compared to control group. And for percent increase in lifespan, our nickel complex have shown 58% more. And the dose was, at the time, the dose was 50 milligram per kg body weight. We have also checked the hematological parameter, that is RBC, WBC, and hemoglobin. And in all cases, our nickel complex have shown very promising result, and all the values goes to normal control. We have also checked the liver function test and kidney function test, and we have checked ALT, STLP, bilirubin for liver function, and urea and creatine for kidney function. In this case, our nickel complex have shown a very good result, and it also goes to near to normal control. And finally, we have checked the histopathology of liver and kidney, and our complex treated liver have shown no complexity when compared to normal liver. That is normal central vein. There is no hepatic sinusoid, and for kidney, intact Bowman capsule, and there is no hemorrhage when compared to normal kidney and normal liver. And also it is also proved, it was also proved in that data. And finally, we have checked the cell cell morphology by fluorescence microscopy and optical microscopy. And in fluorescence microscopy, cells from control mouse demonstrate regular and round-shaped nucleus. That is very clear. And cells from treated mouse, that is nickel-treated mouse, demonstrate irregular and deformed-shaped nucleus. Here, the picture is very much clear. And in optical microscopy, cells from control mouse demonstrate regular and round shape, and cells from treated, that is nickel treated mouse, demonstrate irregular and deformed shape. And finally, we have RNA was extracted by using T100 RNA extraction kit and run into 1% agar gel to check RNA quality, and concentration of RNA was measured using nanodot 2000. And finally, we have checked the expression pattern of our apoptotic genes, and in all cases we have got very clear result and very good result. For GAPDH, the housekeeping gene Caspase 3, 8, 9, P53, PAX, BCL2, cytochrome C, which are apoptosis related gene, and our treatment group, that is nickel treated group, which gives the prominent result. And this is another apoptotic genes, NF kappa B and TNF alpha. In that case, we have got the promising result. And DNA fragmentation is another biochemical hallmark of apoptosis, and our treatment group, that is nickel treated group, gives the DNA fragmentation. And finally, we have checked the in vitro study, that is ESC cells and MCF7 line. MCF7 cells, two cell lines actually used, and MTT colorimetric assay was used, and in both cell line, ESC cells is mammary gland cells, mammary gland cancer cells, and MCF7 is breast cancer cell. And we have studied our nickel complex and highest concentration 500 microgram per ml in ESC cell lines. It has shown 76.4% cell growth inhibition, and MTT and MCF7 cell line in the highest concentration 500 microgram per ml, it has shown 59.7% cell growth inhibition. And the total picture of the work is the complex showed potent anti-hemolytic activity. MTTSS showed dose-dependent anti-cancer activity. The complex inhibits the growth of cancer cells above 51.81% and 75.75%. Complex restores different blood, liver, and kidney parameters. Significant morphological changes of cancer cell in response to the treatment with the complex was observed using optical and fluorescence microscopy. DNA fragmentation assay clearly demonstrated the evidence of the apoptosis of cancer cell. Level of expression of cancer-related genes P53, BAX, BCL2, Caspase 893, NF kappa B, TNF alpha, and cytochrome C were measured semi-quantitatively. Using all the data of gene expression and DNA fragmentation, we have constructed an apoptotic pathway of cancer cell growth inhibition by our newly synthesized complex. And this is the overall pathway for our newly synthesized complex we have constructed. So I am very thankful to the Ministry of Science and Technology of Bangladesh for giving me the financial support, the authorities of Institute of Biological Science in University of Rajshahi, Bangladesh, and Ruhul Amin, Senior Scientific Officer, BCSI Laboratories, Bangladesh, for providing me nanorock facilities, and other person who have joined with us. And finally, we have published this data in Applied Organometallic Chemistry 2020. So thanks to all. This is our lab member, my supervisor, and that is our professor, MT Abureja, me, and our lab mates. So thanks to all.